This protocol allows the simultaneous modulation of many microRNAs in eukaryotic cells and is based on a very simple and flexible method, which minimizes molecular cloning steps. This technique has implications for the study of microRNA functional interactions in vitro, but more importantly, it provides a platform for a more effective use of microRNAs in therapy. We have mainly developed and used it in the context of brain cancer, but it applies to any diseases where multiple alterations of gene expression are involved.
We have tested our artificial transgenes in other models, such as breast and thyroid cancer lines, showing the reproducibility of this technique. Basic knowledge of bioinformatics and microRNA biology is helpful for this protocol, but not indispensable. A bioinformatics approach is important to define relevant microRNA combinations at the theoretical level, while familiarity with the microRNA processing machinery is important to understand how these different microRNAs can be assembled into artificial DNA clusters that can be successfully processed once delivered to receiving cells.
To coat dish, first dissolve poly-D-lysine in water at a concentration of 100 micrograms per milliliter. Pour the solution into a dish at the amount of 1 milliliter of solution per 25 square centimeters surface. Swirl the dish to make sure of coverage of the whole dish. After 6 hours, aspirate the poly-D-lysine solution and rinse twice with PBS. Plate human neural stem cells in the amount of 100,000 cells per 5 milliliters of medium, either in stem cell medium, astrocytic differentiation medium,